everyone, welcome back to our channel SS Biology. Today's topic is about enzyme and its active site and how enzyme works in our body. So let's begin. Before we begin, we need to know what is an enzyme. An enzyme is a thermolabile proteinaceous, pH sensitive organic catalyst, that are synthesized by the cell and may catalyze a thermodynamically stable reactions inside or outside of the cell and remain unchanged even after the catalysis is or the reaction is done. So from here what we get that protein is enzyme is a proteinaceous compound. That means it's a protein. It is pH sensitive. Second one is pH sensitive, and thermolabile. So what we get from the definition: first, it is a proteinaceous compound, means it is made up of proteins. Second, it is pH sensitive, means the concentration of H plus plays an important role in the enzymatic activity. The third one is that it is thermolabile, means the temperature also plays an important role in their enzymatic activity. How this pH, the concentration of H plus and the temperature plays an important role in the enzymatic activity, we will discuss it later. As enzymes are made up of proteins, so they are made up of amino acids. If an enzyme is made up of single polypeptide chain, then those enzymes are called monomeric enzyme, and if the enzymes are made up of more than one polypeptide chain, then those enzymes are called oligomeric enzymes. Examples are being lactate dehydrogenase, pyruvate kinase, etc. Enzymes are often associated with a non-protein part which is called the prosthetic group or coenzyme. So in an enzyme we get a protein part which is called apoprotein and a non-protein part which is called prosthetic group, and together this prosthetic group and apoprotein is called holoprotein. The enzyme works by binding to its substrate at a specific site which is called the active site, that is mostly present on the surface of the enzyme. This active site is highly specific for each substrate, which means the active site only can bind with a specific kind of receptors or the specific kind of substrate, not the all kind of substrates. That's why different kinds of enzymes are present for the different kind of substrates. Now previously I have already talked about the enzyme is made up of proteins, which means it is made up of amino acids. When protein is first synthesized after the translation, that protein is called naive protein and it undergoes the protein folding to get a functional structure. This folding is very important. Unless the protein is properly folded to its tertiary structure, it does not gain its functionality. If the protein or the enzyme are misfolded or improperly folded, that protein or the enzyme will become a faulty protein that will not function properly. This proper folding is needed to form the active site. Let us talk about the example of chymotrypsin. The functional form or the tertiary structure of the chymotrypsin contain 241 amino acid. The proenzyme form of the chymotrypsin contain 245, but the functional form of the chymotrypsin contain only 241 amino acids. So from this data we can get that it is a large enzyme. The chymotrypsin contain 3 chains: A chain, B chain and C chain. A contain 13 amino acids, B contain 131 and C contain 97 amino acids. Out of which they contain 31 aspartate, 27 serine and 2 histidine in their structure. But the active site is only constituted by 1 aspartate, 1 serine and 1 histidine, and their position is what? 102, 195 and 57 respectively. So from this data what we get that even though enzyme is a large molecule, the active site is relatively small. Not only relatively small, it is very small. It is only constituted by 3 amino acids. Also, when they are first synthesized, these amino acids which constituting the active site is not closely present. When they are first synthesized, these 3 amino acids are not closely present. They are present wide apart. But when they are being folded, they come close to each other and form the active site. Let's suppose take that example. When they are first being synthesized, this is a naive protein. The aspartate at 102 position, serine was at 195th, and histidine as 57. When they are being properly folded like this, these 3 enzymes comes close to each other. This one is histidine, this one is aspartate, and this one become serine. 195, 57, and this one is 102. So this is the enzyme, and this is the active site of the enzyme. Active site. This active site is highly specific. Another important term in enzymatic activity is activation energy. The activation energy, the minimum amount of kinetic energy that is required to produce the product from the substrate. That is the minimum amount of energy which is needed, to produce the product. Okay, that minimum amount of energy is called the activation energy. Okay. Enzymes always work by decreasing this activation energy. Let's talk about an example. Carbonic anhydrase that catalyzes a reaction between carbon dioxide and water and produce HCO3, bicarbonate and H plus ion. This reaction, if occurs in absence of carbonic anhydrase, the rate of this reaction is about, in absence of carbonic anhydrase, is about 1.3 into 10 to the power minus 1, which is very slow. But if this reaction occurs in presence of carbonic anhydrase, the rate of reaction is increased into 1 into 10 to the power 6. There is a significant increase in the 1 into 10 to the power 6. That is a significant increase in the rate of enzymatic reaction. And how this increase, the rate increase occurs by decreasing the activation energy. That's it, guys. That's all for today. If you have any question about this topic, the comment section is open. Do comment there, or you can directly message us in our Instagram or mail us in our official Gmail account. The next video which I'm going to make about the various factors which is regulating the enzymatic activity. So stay tuned in our channel, and if you like this video, do not forget to hit the like button and share with your friends, and subscribe to our channel for such videos. Thank you.